Hello, students. In this particular session, I'll be discussing about the different errors in metabolism. Now, the paper Clinical Biochemistry has different units which covers errors in carbohydrate metabolism, errors in lipid metabolism, errors in amino acid and nucleic acid metabolism. And then there are two units which covers the liver function tests, kidney function tests, pancreatic function tests, gastric function tests. Okay, so these are the tests which are usually covered in clinical biochemistry as as diagnostic tools. Now, this particular paper which covers inborn errors of metabolism and the diagnostic tools in order to identify these inborn errors are quite interesting, and this is more clear because when we study the metabolism paper, we can actually correlate this particular paper, for example, the errors in metabolism. So simultaneously, when you're covering both the papers, it becomes very interesting. When you're very thorough with the metabolic pathways, whether it be the carbohydrate pathways, which metabolic pathways, which covers glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, HMP shunt, and so on. And whether it be the lipid metabolism, whether it be the alpha oxidation, beta oxidation, omega oxidation, or the synthesis of cholesterol, or catabolism of cholesterol, and so on, or the sphingolipids synthesis, and so on. And if we discuss in detail about the different anabolism of different amino acids, catabolism of amino acids, proteins, nucleic acids. Now there are so many enzymes involved in all these metabolic pathways. God has made us so beautiful, and all these enzymes are working in unison and coordinating with each other, and we are all leading a very healthy and peaceful life. If a particular enzyme, any small enzyme, is defective, it can lead to, it can be a very small problem, or it can lead to fatal problems. Okay, a defect in an enzyme. So as you learn inborn errors in metabolism, you start actually, admiring God Almighty and thank, and we become more grateful to God for making us so beautiful with thousands and thousands of enzymes, thousands of molecules in our body, communicating between each other, sending cell signals between each other. There are there's beautiful communication between the cells so that everything works fine. If a small defect is there in the synthesis of the particular enzyme, if a small defect is there in the gene for synthesizing a particular enzyme, that can lead to a metabolic defect, or a full stop in the pathway of the synthesis of an enzyme, or the breakdown of a particular product, or the synthesis of a product. So even a small defect in an enzyme can lead to either an accumulation of a product, or a defect in breaking down of a product, which can become toxic. Okay, so there are different disorders connected with the defect in either enzyme or particular receptors connected with the enzymes in the metabolic pathways, and these are inborn errors of metabolism. Now there are a number of diseases which are there in the inborn errors of carbohydrate metabolism. For example, the very familiar diabetes mellitus, hyperglycemia, okay, and the other lipid metabolic errors, like, for example, hypercholesterolemia, hypolipoproteinemia, hyperlipoproteinemia. Okay, and like that, we have many in the amino acid and nucleic acid metabolism also. For example, cystinuria, okay, uricemia, and porphyrias, and things like that. Okay, so these are all covered in this particular paper, which we are dealing with the clinical biochemistry. And in the end, we will be having all the different functionality tests of the liver function test, the kidney function test, the gastric function test, okay, and the pancreatic function test, how these tests are carried out. And how do you arrive at it? What are the normal values? If there is a difference in the normal value, what does it signify? Okay, so these are all covered. Thank you.